Here we'll learn about DNA damage and repair. To begin, start a table to learn some key mechanisms of DNA damage. Denote that endogenous or exogenous agents can induce DNA damage. For endogenous damage, denote that spontaneous chemical reactions can damage a nucleotide or nucleotides. For exogenous, denote that damage can be initiated by exposure to mutagens, chemicals, or radiation. Denote that spontaneous DNA damage is most frequently in the form of deamination, in which nucleotides lose amine groups, depurination, in which a purine, adenine or guanine, is released from DNA. Denote that mutagens can cause damage in the following ways: the formation of pyrimidine dimers, which is induced by UV light exposure; alkylation, the addition of methyl or ethyl groups to nucleotides; and the addition of a bulky group, which often occurs upon exposure to carcinogens, cancer-causing mutagens. Let's illustrate these forms of damage now. We'll start with endogenous DNA damage, deamination, and depurination. First, deamination. Draw a base as a hexagonal ring and specifically label one carbon. Show it bound to an amino group. Label this base as cytosine. We won't worry about the rest of its Lewis structure. Next, draw the purine adenine as a hexagon bound to a pentagonal structure. Again, label one carbon in the hexagonal portion bound to an amino group. Now redraw both cytosine and adenine without their amino groups. Instead, replace them with a carbonyl oxygen. Show that the deamination of cytosine produces uracil, and that the deamination of adenine produces a molecule called hypoxanthine. Now draw the sugar phosphate backbone bound to all four bases. Deamination only affects the nitrogenous base of each nucleotide, not the backbone. Write that deamination can affect DNA replication and gene expression if not repaired. A single nucleotide change can alter the codon and corresponding amino acid. Now for depurination. Draw a double-ringed purine and label it adenine/guanine. It represents both purine nucleotides. This time, show it attached to the deoxyribose of the sugar phosphate backbone, a 5-membered ring bound to a phosphate group. Next, illustrate that the bond between the deoxyribose and purine spontaneously cleaves. Redraw the deoxyribose without a purine bound. Label it an apurinic site. Like deamination, depurination does not break the sugar phosphate backbone. Finally, let's learn exogenous damage, pyrimidine dimers, alkylation, and bulky group addition. We'll start with the pyrimidine dimers. First, write that they're caused by exposure to UV radiation. Now draw a DNA strand with a 6-membered pyrimidine ring and label carbon 5 and 6. Show that carbon 5 and 6 form a double bond. Indicate that carbon 5 is bound to an R group, which is just a hydrogen in cytosine, and a methyl group in thymine. Indicate that carbon 6 is bound to a hydrogen in both pyrimidines. Next, draw another pyrimidine adjacent to this one, labeling the same atoms. Now indicate that the carbon 5 and 6 of one pyrimidine binds the carbon 5 and 6 of the second pyrimidine. Indicate that the resulting structure forms a cyclobutane ring. Write that these cyclobutane pyrimidine dimers occur only between adjacent pyrimidines, most often thymines. Write that they distort the DNA double helix. As a clinical correlation, write that these distortions can produce skin melanomas. Now for alkylation. Draw a methyl group, CH3, and an ethyl group, CH2CH3. Indicate that these groups can add to DNA bases, both purines and pyrimidines, at numerous positions. Finally, bulky group addition. Right, that this mostly occurs upon exposure to carcinogens, which are mutagens that make cells cancerous. Draw the bulky group benzoapyrene, an aromatic polycyclic structure. Right, that this structure can react with both purines and pyrimidines at numerous positions. Right, that these bulky groups also cause distortions in the DNA double helix. As a clinical correlation, write that carcinogens in cigarette smoke form covalent bonds with DNA and disrupt hydrogen bonding between nucleotides. This distorts the helix and causes a frame shift that changes the character of all the subsequent codons in the DNA strand. Constant exposure to carcinogens can lead to cancer, most commonly lung cancer, when it comes to cigarette smoke. To emphasize this point, return to our table and denote that DNA damage can increase the frequency of mutations, which include nucleotide substitutions, deletions, and insertions. Now that we know the most common types of DNA damage, let's add the major repair pathways to our diagram. First, write that mismatch repair fixes the replication errors that are missed by DNA polymerase's proofreading and exonuclease activity. Write that it cannot repair damaged DNA nucleotides. Then indicate that the base excision repair pathway can repair deamination, depurination, and alkylation. Finally, indicate that the nucleotide excision repair pathway can address pyrimidine dimers and bulky group addition. We learn the mechanisms of these repair pathways elsewhere. This concludes our diagram.